Subscribe to our YouTube channel and press the bell icon to get the latest updates.
Today we're coming to you from the CSIR Center for Cellular and Molecular Biology, primary research institute working on the COVID vaccine here in India. To know more about what's going on behind the scenes, we're going to be speaking to the director of the institute, Dr. Rakesh Mishra. Straight away, Ccmb, as we all know, has recently declared that it's one of the institutes to have discovered a clade that is perhaps one of the most prevalent as far as the virus in India is concerned. If you could please elaborate about that.
Yeah, so we have been sequencing the genome of the isolates from the patients in India, and many other labs are sequencing. And what we found is that in India there are two major clades. One is known, which is world over major clade called A2A, and we found a new one, a distinct clade which is unique to India and Southeast Asia. We call it A3I, and this is second most dominant. It makes close to 41% of the patient infection in the country. So A2A and A3I put together are more than 80% of the infection here. Interesting thing about this clade is that this is first of all unique to Southeast Asia, so it has not gone beyond. It also tells us that when the infection originated from Wuhan, then it came to Southeast Asia, and that is where somewhere in mid-February this new clade emerged, because we can calculate back the timing based on the mutation rates, and there are specific techniques for that. In fact, we can calculate back and say that sometime between mid-November to mid-December, the initial virus originated in China. So what happens then is from Southeast Asia, this virus came to India, most likely to Hyderabad, and that is from where it has spread.
Oh, so the first place was Hyderabad, as has been this thing.
Before that, from Wuhan directly, some infection has come in Kerala, but that were isolated cases and it was contained well. Second major one came from Hyderabad and it spread in Hyderabad, in Telangana and Tamil Nadu, and there is some part in Delhi also, because we are talking about late March, early middle March or something, where the lockdown was not there, so people were still moving. So maybe that is how it was spreading. Interesting thing of this is that in addition to that, now it divides our patient into two groups, so we can study them separately.
And could you please elaborate on what are these groups for our viewers?
So the clades are defined by mutations which are characteristic of these clades. That's how actually they are computationally defined. So for instance, A3I has 4 defining mutations in it, out of which 3 are what we call as non-synonymous, which means the protein sequence also changes, and 1 is synonymous, which means protein sequence remains the same. One of the mutation is in the RNA-dependent RNA polymerase, RDRP gene, which is gene required for virus to replicate and produce more of these transcripts and proteins in the cell when it infects. So what we see is that this particular mutation, which defines one of the 4 mutations define this clade, is in this gene, which our computational analysis shows that should make the protein less efficient. Theoretically, this needs to be validated, this needs to be correlated with the patient, but this makes us think that this particular clade might be slow spreading or slow growing, and therefore may be less symptomatic in the patient.
So that's an important point that Dr. Mishra here makes. That is that it might spread at a slower speed, which could be comforting news for people in India, that means.
Yeah. So this will be certainly, that means it will not be
it won't spread at the pace that one was expecting, because the virus is known to be very rapid, if I'm not wrong.
Yeah, so in this case, the virus will be less efficient. So it may spread less efficiently, it may be less symptomatic. I mean, either of the two can happen. It is early stage, none of both can happen as well. But now we have way to investigate it. Because now we go to patient, we are collecting samples from patient which have this particular clade. And then we are collecting the data about the patient, how was the diagnostic prognosis, whether they were asymptomatic, they were from ICU and so on. So I think in a couple of weeks we'll be able to establish whether A3I is less symptomatic, less aggressive, and less slower in spreading. We can make out that now.
Right. And also when you talk of the two groups from which you're collecting the samples, so correct me if I'm wrong, one category is the asymptomatic patients and the other is which has mild symptoms, if I'm not wrong, to draw the comparison?
So we are collecting actually all kinds, but we will comparing those who are in the ICU, so which means they have severe symptoms, and those who are either mild symptom or asymptomatic. So these are the two, and our guess is that maybe two different virus clades are making two kinds of thing. That is just a speculation. It could be that differential response is because of the difference in the people, not in the virus. That also is possible. So that is what we are now very keenly investigating, and in next few days, in a couple of weeks, we'll figure it out.
Right. Before I move on to, the science-related questions, of course, since we're talking about testing, that continues to evoke a lot of debate across the country, with the ICMR guidelines saying that, not to test asymptomatic or to test, if I may say, only symptomatic patients. We've seen what's happening in Delhi. In fact, sitting in Telangana, I can confidently say that it's been grabbing headlines for not testing enough. Is it not important to test asymptomatic patients to understand the gravity of the situation?
See, the testing is like seeing what is there. If you see more, you will be more informed. Now you have to draw a line how much we can test. It's because if you gave me a choice and resource, I will test everybody, because that will give me the best information, best position to decide where to do a lockdown, where to restrict movement, where not to restrict, where to open business, where not to open business, who are the people who should be sent to quarantine, and so on. And then if we can do that effectively, it's a matter of 2-3 weeks, we can really control the situation. But easier said than done, because people move, it's a free country, there are lots of people ignoring many of these things. But in any case, if we can test more, we'll be in a better position. But it's a government may have other restrictions. I'm not going into that, whether they want to test or not, whether they don't want to tell data. I don't think that is, I'm very keen to discuss that. But what government may be having limitation is because testing is expensive, which means you have to divert your resources of healthcare workers, because those are the ones who are going to collect the sample. And then you need trained people to do testing. Each test is expensive, 2,000, 3,000 private places. If you add more running cost, it could be 3,000-4,000. So it is expensive business at the moment. So ICMR doesn't say that don't test asymptomatic. They are saying this test symptomatic, that is necessary, but asymptomatic, somebody has secondary contact, somebody is something, or for any reason, or I would say that healthcare workers should be routinely tested every 2 weeks or every month or whatever. So, but in the end, it boils down to whether you have resources, whether you have efficient system. Can we do that much?
Since you mentioned, and in fact, that's been the common defense that most of these state governments have been taking, that is the limited resources that they have. Ccmb here has been working on a modern technique to come up with cost-effective way and enhancing the speed of the RT-PCR tests, which I think can be quite effective. Could you shed more light on that?
Yeah, so one of the things, when we started testing, most of the testing, even now, majority of the testing is done by our PhD students. So these are bright young people. They are not just doing simple testing. They are constantly thinking, can we improve it? Where is the bottleneck? Where is the problem? So by doing some working for a month, figuring out the problem is, we have sorted out few things. One is, we worked out the way that we can do pooling of the samples. If the occurrence is less than 2-3%, less than 5%, you can do pooling of the samples. So we tested some and figured out the sensitivity and all that, and they suggested to government, and the ICMR also came with the advisory. And in fact, we have been pooling samples in Telangana. So 5 samples in one pool. So which means you can do 5 times faster and 5 times cheaper testing. So that is one thing. But
And that will not impact the outcome or the test results for that matter.
so, as I mentioned, if the occurrence of the infection is less than 2-3% or 4%, it is safe. But if it is more than that, then it will not be very rewarding strategy, because if you are testing 100 samples, make a pool of 5, so you will test 20. If 1 or 2% were positives, then 1 or 2 pools will show positive. So you have to go and test another 5 or 10. So that means in 20 plus 5, 10 means up to 30. If you do, you can give the results of 100 samples. So that is a good one, 3-fold extra you got. But if the positives are 6%, 7%, then you have to go and do 50, 60 one more time. So then it is not at all economical to do pooling. So pooling of 5 in one pool is good only when the occurrence is very less, 2%, 3%. And we knew there are 12, 13 districts at that time in Telangana who had low occurrence, and we were pooling the samples. And even now samples come from there, they will pool, because we know the occurrence there is much less. So that was one thing we did. But more important improvements that our students have established now is that samples are collected in swabs, which are put in a solution, a pink color solution called VTM, viral transfer medium. This 3 milliliter of medium is used to dip the swab, and that liquid, that swab is VTM is used for transporting from the collection point to the testing point. Now you are transporting liquid, it leaks. So you have to seal properly when you take out, and your sample is now diluted in 3 milliliter. Right. So if the viral load is less, then from 3 milliliter, actually you take a fraction of a milliliter for testing. So that means sample is very diluted, many times it can be negative. So we have suggesting that, which we have established and shown, we have sent our recommendation to ICMR, that don't use VTM, take directly swab and transport the sample in dry swab. So the dry swab, it's a dry sample, there's no liquid, so handling is very easy, it's very safe, people can open, it will not leak out, and it's VTM is imported, so you save money. And then when you take out when you reach the test lab, there you add a simple buffer, we call it TE, Tris EDTA buffer, which is very, very cheap, like water. And then you add some 400 microliter, which is a little less than half a ml of that, and leave it for 30 minutes, and you are set. You take a small quantity from there, heat it to 98 degrees, 6 minutes, and then you can go straight for RT-PCR reaction.
So this will definitely enhance the speed from what I understand.
Exactly. So what you are doing is now, in the normal testing is you take the VTM, take out 150 microliter, inactivate the virus, then do RNA preparation. So RNA preparation takes 2.5-3 hours, and one person can do 23 samples, for some reason 24 or 23 samples in those 3 hours. That step is eliminated. So you straight away take the T and go straight by one heating step to RT. So not only you save the time, so 3 hours is this time, and the rest is actually 2-3 hours you do rest of the things. So 50% saving in time. And if you are not doing RNA preparation, that reagent is also expensive, you don't have to spend. Each reaction costs some 300-400 rupees for the RNA isolation. So that means you are going to save something like 30%, and VTM cost you at up close to 40% cheaper.
I also want to try and understand one more thing, since CCMB is of course closely observing the virus, handling with it. What stage has the vaccine reached at? Can we expect any good news in the near future? It's something we're all very curious to know.
Vaccine, actually whole world is very keen to have a vaccine, because vaccine means you will not have the disease, and we are safe, because so many like polio and all we have fought well, and they are out. Drug is second choice, because drug means you have got infection, you fall sick, go to hospital, now you need the drug. So everybody wants vaccine, and in fact in the history of the world, this is the first time that for one disease at one time so many companies and so much effort and money is being put. So with so much effort, technology at this level, we expect results. Having said that, you are trying for something which has not been achieved before. You are trying for an effective vaccine against a coronavirus, which is not there so far. People have put so much of money, we don't have vaccine against HIV till today, 4 decades of effort. But that doesn't mean that we should discourage. This is possible that we'll get the vaccine. But the vaccine, even let's say today we have vaccine candidate. There are some cases where the first trial was good, that is safety was good, so it is acceptable. But you are talking about multi-stage trials in which some smaller number, then few hundred, then maybe few thousand people are vaccinated. Then you wait that even in normal condition they don't get infection, or you give them the infection. You see how tricky it is that in unproven vaccine, you have to have volunteers who are ready to act. So there are many difficulties, and all these, each stage takes couple of months, and collecting data, analyzing, going to the regulatory authority again. So you're talking about 4, 5, 6 months of simple work of this kind. Then even if the vaccine comes in place, then you have to go to production. You need billions of doses. We will not be the first one to get. First one, those who are investing money, they will get. Now government India is also putting money. So for the same reason that we should get vaccine as one of the first. But production at a large scale, then supply, all those things are going. So we are talking about in best possible scenario, maybe first half of next year, not before that.
Since the institute is so closely working, on the coronavirus, and of course we will be moving to the biosafety lab, which is where labs can actually test or study the coronavirus. But to move to the next question, it's not very often. It's a challenge to culture the virus, from what I understand, given that the human virus is not very long lasting in a lab. So how are we handling it here? Is there an immorality scene violence that we're using? There is something called the virus cell, which I understand, which is taken from the kidney of a monkey, if I'm not wrong.
Correct, yeah. So growing this virus requires human cells or mammalian cells. So any virus by itself, it is a particle which is waiting for something to come, then only it can. So you need somewhere to infect this. So and coronaviruses are known to be grown in Vero cells. So Vero cells are African green monkey kidney epithelial cells. They are established cell lines in which you can grow the virus. A few tricks in this. One is that this virus is so contagious that you have to work in biosafety level 3 laboratory. Many places don't have this kind of facility. So we have done that using Vero cells, and we are also using now another version of the Vero cell which is called 6E, which is little better than the Vero cell. And interestingly also, we have gone in collaboration with a company in Bangalore called iStem. They have human epithelial cells from the lung, which is exactly where this virus infects. So that is the best system to have, and those are the cells which they have made by some treatment, what is called iPSC, induced pluripotency cells, and those are the kind of stem cells. They can keep growing for longer time. And if you infect the virus in that, then you are having very close as one could imagine to the human situation, because now you are having not monkey cells, you are having human cells, you are having lung epithelial cells. That's how the virus goes. We cannot do experiment with the people, but when we have in the cell system virus growing, then we can study the biology of the virus. So that is for us most important thing. If we know more, we will be able to manage it better. The second thing we can do is, now since the virus is growing in the cell, and the more and more virus is produced, the cell is dying. Now you come up with some drug which is anti-COVID, which will stop COVID. So this is, now you can test a large number of drugs and see which one is working. That means you have got something which is very useful, very promising. Now you go to patients. So otherwise there is no way of, theoretically if you predict something, you cannot go and inject in the patients. Right? So this gives a very good handle to do a primary, what we call as in vitro screening for the potential drugs. It could be a drug, it could be a phytopharmaceutical, it could be anything. So, but we have system where we can, we can test them using this cell culture system other than studying the biology.
And I think now we'd like to move to the biosafety lab, or at least outside. Of course, we're not allowed inside, and we respect the privacy, of the scientists. But just to get an understanding as to how the team here, which I believe comprises about 60 people, 45 of whom are students. So that and a lot more right here on the campus.
This is a BSL-2 setup. What we are mentioning is a BSL-3 setup where we can handle the live virus. Then virus can be grown in the cell culture. For that we require different negative pressure and different kinds of hoods. So that is where we grow the virus. But if they only for testing the virus, as I said, will be brought inactivated here, and then RNA is isolated and ready for the RT-PCR. As you can see from this pass box, the samples come from outside, and then back sample is brought from here, then taken to this RNA isolation room. So this is the inside we do the RNA isolation. The sample will go inside, and the sample will be handled in this hood. These are biosafety level 2 hood. Here sample will be brought and RNA will be virus will be killed in the RNA isolation or inactivation buffer, and then with the help of kit we process and put the thing in this machine which does the RNA extraction. So this takes 2-3 hours, this whole process, and then once RNA is extracted, then the sample is ready for RT-PCR, which will be in the next room.
So how many samples can we expect to be tested in one?
A center can collect up to 1,000. More than 1,000 a center will not be able to do because you require certain logistic. We will collect samples from 20 such centers. All those 10 or 20 or 30 or up to 50 can come to one place. So we are talking about 20, 30 or 50,000 samples that reach in the evening to the NGS center. There we will make the library and put it for in the NGS machine, and it takes 7-8 hours, and then it will give the results. So and because the samples initially were barcoded, the next day we will be able to figure out which one of those 20,000 or 30,000 samples are positive and which are negative.
So that's pretty interesting. You're saying if this module is successful, then we could actually do 50,000 tests in one go.
Yeah, 50,000 in one go, but that test will take 2.5 days. Okay. But we can
even then,
we can put it in a train that everyday machine will be running, so we can. So result will come after 2 days.
Okay.
So this will require a logistic that, let's say if you imagine a Hyderabad, then all over the Hyderabad, neighboring places, we can collect these small centers where we can have 500 or 1,000 samples. They process. They by evening they send the sample by a vehicle here, and we make the library. Next they run the machine, and following day we give them the result online by decoding for each patient specifically.
And also, sir, of course, here we've been allowed to come in, with our little equipment and gloves. But if we go to the other lab where you said the virus is alive, of course it's contagious. So what are the protection measures that you all as scientists take as well? Are these PPE kits different from what frontline health workers wear?
No, we have the same PPE kit what frontline workers will have. So they are double layer protection, and then you have a mask of different kind, the glass and the shield and a head cover. And those are the things once used they will be thrown. Here this lab jacket we can use again, but there like gloves everything is thrown again. And when you enter you have to disinfect things. You cannot go in and out. It's entry protected. There's a different level, and more important is that it's always under negative pressure. So which means anything from there will not go out. Everything will come and air is sucked through a HEPA filter so that whatever goes out is filtered and is safe.
And also, sir, just to understand, how is the vaccine that's being developed here different from, say, the one at Oxford and the Serum Institute?
See, there are many different kinds of vaccines. So what Ccmb is involved in is interactive with company where we make large amount of virus and then inactivate that virus, test that is inactivated, then it is safe, then it is given to company. They will prepare in a formulation which can be used as a vaccine. And try first in an animal, then some trial, some in a few patients and so on, and then go. Once it is working, then they can take the virus culture from us and do large scale production and go for vaccination program. The other ways of making the vaccine are that you take the specific protein of the virus, clone it in the mammalian cell system and produce that protein only, not the virus, only that protein in large quantity, use as vaccine. There other Oxford I think is doing the RNA based vaccine. So they inject the RNA which will inside the body will make the protein and will vaccine.
While we are not doing that.
We are not doing that. We are taking the whole virus, boiling it, heat inactivation and then giving it for as a vaccination. So there are multiple, there are 4 or 5 different ways of making the vaccine. All should be tried because you do not know which vaccine will be good for which age group, which ethnic group, which virus strain. So there are many variations in this. So multiple option in a, at least I think we should have 5-6 different successful vaccines so that we can take care of this disease on world level.
So while of course we got a flavor of what it is like when these samples are collected and tested here at the Ccmb, but last not the least, sir, my question, of course it's still a couple of months away, that too in an ideal case scenario if we have a vaccine. To all our viewers out there, what would you like to say in terms of ensuring social distancing measures? And we've also seen a massive rise in the number of cases ever since India has started unlocking.
I think that is very important. We are not couple of months. We are, if at all, 6 months, 8 months away from vaccine, if at all. We might not get vaccine even then. So today, considering that we don't have a drug, we don't have a vaccine, so, and this virus is very fast spreading, is very contagious. But there is something which is comforting in the sense that there are certain things which we know. What we know is that how this virus spreads. It's always person to person. There is no other way. So if we can block that, actually we can win. And there is very simple to block that. Always wear the mask when you are outside. Keep distance. 80%, 90% of the infection is because directly from the breathe or speech or cough and goes directly to the other person. So if you are wearing the mask, you are almost neglected, taken care of that. And if you maintain the social distancing, we are almost sure that you are not going away. The other way is that when we are talking, when you are moving around or somebody sneezes, the virus particles in droplets can come in objects like this. If you touch that, then it will come in your hand. And then if you eat something, this is from hand to mouth. So that is why all the time it is said that when you touch something, when you go home or frequently wash your hand with soap. We cannot, the ideal is let people not move, let nothing happen. And then, that is not practical. We have gone through lockdown for 2-3 months and country has seen very bad economic consequences and movement of the people. So we have to learn how to keep social distancing, personal hygiene and this kind of discipline and so that we don't create a crowd in the hospital. See, the 80-90% people will not need hospitalization, but those 10, 5%, 15% who will need, if that 15% becomes in lakhs, then a city like Hyderabad will have problem.
In fact, that's another thing, sorry to interrupt, I wanted to ask. A lot of our viewers keep asking this, that one is, of course, if you've tested positive, but many a times there is someone in your neighborhood who has tested positive. What measures should someone take immediately,
The measures are the same. If neighborhood somebody has tested positive, we have this app. We can if everybody is following that, we will know if how many people and those things that can be used. So we know that in what kind of zone you are. Essentially you have to, if you maintain the distance, if you don't mix with unknown people, and when you touch something, you wash your hand. That's all you need to do. This virus will not fly and go somewhere. It will not going to go and grow in the dirt or in something or mosquitoes. No, none of those things. It is person to person. So there is a good thing that we know how exactly it happens. So we can actually control it. But only thing is we have to be determined.
Discipline.
If we depend on the government that something will happen, if we depend on the drug something will happen and the vaccine, then we are looking in the wrong direction, because that is not where things will come. Things will come from within.
And sir, one last question. Is there any, of course we are noticing it more among elderly people, but in the recent past we've also seen some very young deaths because of coronavirus.
See, the corona, what it does, it goes inside and damages the epithelial cells in the lung, and that creates breathing problems, pneumonia and those kinds of things. When you have this situation, then you have less oxygen supply, heart will now try to work faster, and then most of the body tissue and organs are under stress now. If there is any weakness anywhere, that will flare up. So that is why the death is more if there is another health issue. Otherwise, if oxygen supply is given, ventilators are given, and person gets little more time, the chances that person will recover are better. So in this case, like you just mentioned, if even a younger person has any health issue, it is possible. And of course, there is a condition where they with too much of damage in the leg and person didn't have good immune fight back, it can happen. It is after all a serious disease.
So also, if I may ask, the COVID, virus has also shed light, perhaps on the importance of science in our country, which perhaps was overlooked otherwise. Would you agree? Or scientific solutions, so to say.
Yeah, see, we spend very little money on research, science and technology and development of this kind for many reasons, for economic reasons or whatever. But we should also realize the solution to the problems are in the science. They are going to come out from the lab. For example, now one single virus has played havoc, like every whole world's economy has gone down. Solution will come from the labs. So it's very important, and I think industry, big houses, government, they have realized it. As you may be knowing, Ccmb is a constituent laboratory of CSIR, Council of Scientific and Industrial Research. There are 38 laboratories, and we have made 5 verticals. Every day we have a meeting with the DG, Dr. Shekhar Mande, for 2 hours to review what is happening, what we have to do. And we have 5 verticals which are considering all the labs. One is for surveillance, other is for diagnostic, one is for for drug development, and there is one for medical devices, and the 5th one is for supply and supply chain, because that is also an important component. So in these different verticals, we have got into agreement with big business houses in the country, like Reliance is working with us, Tata Sons are working with us, and as I mentioned, Syngene, a major company that's working with us, and there are many other small players which are involved. So both industry and the government has taken a note that science will provide solution to the problems, and I think maybe that is one of the positives. As you have seen that now there is emphasis on self-reliance. Most of these things which were for diagnostics and all, we are importing, and there is short supply. The whole world need the same thing. You will not be the first one to get it. So now we are making indigenous substitutes to all the components of this, and I think this will help not only for this pandemic, this will help for future, and also it will help facilitating research and development in small institutions or universities, because things will be available locally. So I think we are going to get something. We have suffered a lot from this pandemic, but I think there are some basic changes which will hopefully help us in future in big way.
Thank you so much, sir. I think that's been a very insightful interview. And who knows, we'll be back here maybe sometime in the future talking about a vaccine that's been developed. So thank you so much for taking out time for us and speaking.
You're most welcome. It's a pleasure.